The first session, I'll just give a background. The first session will be Professor Balaji Jayprakash, IISc Bangalore. So please be on time. We will start the schedule soon.
Case of saying, okay, this whatnot, DNA, RNAs, and so forth. If you start by defining memory as an information storage, it's not about the brain, it's not about anything else. It's about just information storage, right? That's what I'm going to stick to. And if you say that, in that perspective, you ask the question of what you want to study. Here, I want to study a particular kind of memory storage. There are very different ways of information storage. I am picking out a small portion of it. I'm going to be studying it. What are the intricacies involved in it? And what ultimately makes us go towards understanding the memory itself. In life, several events happen, right? And I'm going to be talking about these memories of events and facts. For example, you can think of a situation where this person here is having a beautiful moment in a lake, enjoying a nice boat ride, and he wants to capture that feeling or the moment with a camera, right? The idea or the hope is that later in life, you look at this picture, you would be able to rejoice and recollect what has happened at that point in time. Now, until this point, there is no problem at all with that aspect of memory. Where I'm going to be talking, what I'm going to be talking to you about is that how fallacious our memory can be. It's not even can be, it's always, almost always. Even though we take it for granted that I know for certainty this is what happened. Okay, so I'm going to drive across at the end of by the end of this talk. If you kind of take home the message, hey, there is this element of doubt in my own memory, right? I don't want to have a complete trust on this is exactly what has happened. Then I would be sold. Okay? Now, until this point, where you look at this picture and be able to recollect that moment or the event, no problem at all. You have a good memory. There is no issues, no uncertainty. However, days later, months later, you ask the question to either of them, hey, while this event was happening in life, your boat was passing through another houseboat. Do you remember? Said, yeah. Can you tell me if this is, that boat was looking like this or was it like that? That starts the problem. Okay. Your ability, see, these two are very different boats, right? I mean, no question here, right? If I ask you, okay, are they the same boats? You would say, no, they aren't the same boats. They are very two different boats. But the problem is, I'm asking you to identify which of the boat that you encountered at that point in time. That's where the problem is. And there you start to have this uncertainties coming into picture where you are visually, the sensory perception, you are able to tell these two are different boats, but yet the content, you are unsure about it, leading to this uncertainty. Okay, good. This is a very common experience. Okay, let's do a small thought experiment. Okay, we all had breakfast. I assume so, right? Good. No, some of them did not have. Good. You had. Okay, excellent. If I ask you what did you have for the breakfast, everybody will say, correct? True. A year before now, if I ask you the same question, you would start to have doubts about what did I have for my breakfast. You can make a guess, intelligent guess, but the surety with which you will be able to tell is pretty doubtful, pretty questionable. Have I asked that question a year before, you would definitely have said, yes, this is what it is. So it's very common. It's not something that it's not some isolated phenomena that I'm talking about. It's a very, very common phenomena. Every day you face it. But we are unaware of what the consequences of these uncertainties could be in our memory. To understand that, let's look into what leads to this uncertainties themselves. I already kind of gave some glimpses of what are all the reasons why these uncertainties can happen, but let's put it in a firm footing. Number one, when you are talking about these uncertainties, you're talking about when we're talking about uncertainties occurring when you are trying to retrieve a memory, right? When you are actually encoding a memory, you don't think about, hey, is it sure or not sure? Right? That's not what's happening. You're reading a question, you're answer, by hearting a question, answer for the question. That's no problem there at all. The problem happens when you're having an exam, right? Right, given a question paper, right? So you're trying to retrieve. That's when it manifests. Typically, it's not a given, but typically it happens when you are trying to retrieve a memory that is older. Older means it's also what we call it as a generalized memory, like the 1 year before what you had a breakfast, 1 year before what color shirt was I wearing, right? So those kind of questions, right? So that when you're a little older, you don't have the specific details, but it's a little bit more generalized. Okay. It also happens when things are related to each other. Okay. Two boats that I talked to you. Okay. They are not just older, but they are also related, right? Two boats. Okay. They are trying to tell apart. Okay. So these three things are kind of a given. You need to have these things for the uncertainty to manifest. And what effects do they have? And this is precisely the question that our lab is very deeply interested in. We believe this can have a very good value in terms of brain function. What that function is, we will come pretty much towards the end, perhaps. But this kind of sets the tone for our lab's research and one of the lab's research. And we are very much interested in understanding what could be the neurological origin, what are the why have these uncertainties themselves, and for that, trust me, you can actually use mouse as a model, okay, to study this. Okay, it has several advantages. We will see. We also do some human studies. Okay, I won't have time to talk about the human studies, but a lot of this can be understood using this mice. So I'm going to stick to that. Okay. All right. What do we do? First thing is to say that, okay, can the mice mimic all these things, correct? Meaning uncertainties, right? How do we do this? The way we do that is, again, many different behavioral paradigms we use, but today I'm going to talk about one kind of a behavioral paradigm and use that to define a notion called related memory, right? Two boats. Go back to that, right? So what happens is that the mice, if we take a mice and put it inside a small chamber, okay, just like we are all coming into the lecture hall and then trying to listen to the lecture. Except now, after being in that chamber for a little bit of a time, I present the mouse with a foot shock, mild foot shock. Okay, this is an aversive event. Nobody likes to have any aversive experience. So what happens is that next time I take this mice and place it in the same box. Okay, the mice starts to express a behavior. Behavior we call it as freezing, wherein it stops moving, stops everything except for breathing. Okay? By measuring how much of the time the mice spends like this, you can actually tell how well the mice has made that memory. Okay. Now that's the kind of graphs that you'll be seeing, the bar graphs you'll be seeing. Each individual dots represent a mice, and that's the baseline. Okay, before the start of the experiment, before you giving the shock, that's the kind of freezing that they do. They just keep moving around all over, right? Now, the moment you give the shock, and then the next day, you can also do it immediately, right? But here I'm showing you the data for the next day. Then what you come back and put them, they should exhibit some kind of a fear. It's a fear response. Why it is a fear response? How well can you say that it is a fear response? All that is are good questions. I have answers to. We can talk about it perhaps later towards the end of the talk. Okay, but just take it from me that you, by measuring this freezing, you can actually tell how good the memory is. This is good. You're putting back into the same box. You have that memory. You can actually alter it. How do you alter it? The way it looks, the way it smells, the way you can alter these things. The setting of the context changes. Okay, just the way lecture hall has a general setting, right? This particular lecture hall has a specific setting. So context A has specific things and context B has its specific things. Both of them are still a context. Like that, you can go to context C. Only difference is A and B are related to each other, meaning very close. They're like the houseboats that I showed you. They're pretty close to each other. But the boat that they actually went on is very different, right? It's not a houseboat at all, right? So same way, context C is very different. Now, how do you know this? Because if you look at the freezing exhibited by the mice, the behavior is very different, right? So this guy shows the highest freezing, very strong fear for the training context, lower and lower. Okay, this 24 hours, 24 hours, 24 hours later, no problem at all. So you have established A, B, and C. People have done it for ages. We are using this. Now what does this have to do with relatedness, right? I kind of told you here. I can change a little bit, make it look similar. Okay. Now, what is the test for it? The test for it is you wait for about 30 days, equivalent of me asking you a year. Okay. You can also do that for 30 days. I just exaggerated for a year. Okay. In mice, we do it for 30 days. Put them back. Now, look at what has happened. What was B, the differentiation between A and B is kind of lost. Just hold on to this. We have much more detailed data of this that's coming up in the next slides. But the point that I want you to take across is that if you wait for 30 days, the ability to tell the difference between A and B is kind of lost. However, they still hold the ability to tell the difference between the C context, and the rest of them is holding good. Okay. So this is important, right? If you were to generally express fear for everything, then it's like anxiety disorder. We don't want to have that, right? You want to have some amount of fear. It's okay, but that has to be driven specifically by the cues and not for anything else. Okay, good. So now this allows me to define what is a related context, right? What I'm going to tell you that a context, initially the animal is able to tell the difference between, but after a certain period of time, it loses its ability to do that, right? Again, if I had asked that couple, is this boat A or boat B that you actually saw it, the next day, they would be able to tell. Just I chose to ask them a year later or a few months later, they're having trouble. Same here. If I ask a day later, A and B can be nicely told, but not a month later. Okay. Now that's one way of testing what is related. Okay. I haven't still come to the point of uncertainty, and in science, you don't just want to take from one way of thinking, right? You also want to look at multiple ways of looking at it and be able to see if there is an emergence of convergence, right? That things have to converge. If they don't converge, there is something wrong. You don't feel comfortable. So what do we do is that we are looking for, is there a molecular means of actually saying things are related? Okay. What do you mean molecular means? Thanks to tons of research, we know that generally memory, right? Memory, if you want to form a memory, there's a class of molecules called receptors of a particular kind called as NMDA receptors. Don't worry about the name, it's just a tag. If they are blocked, somehow you do something to make them not active, then you will not form a memory. Very useful. Why? Any idea when will you use that? So you get hit. Because you have to, or you have to undergo a surgery. Okay, or you have to go through an endoscopy or any invasive procedure, right? You go, the doctor would actually prescribe you some medicines, but the endoscopy is a very good example where he won't completely knock you down. Okay, it's like you are half there, half not there. Okay, and if somebody is going through or somebody has gone, please do this test. It'll be wonderful. You keep talking to them, they would be able to remember pretty much everything until the point a drug has been infused. They will engage with you nicely too, provided the tube is not gone inside. But after the surgery, after the procedure is over, the drug wears off, you ask them, I vaguely remember something was going around like this, but actually don't know anything beyond that. That's very good, right? Because you don't want to have this bad memory of how deep it was. Okay? So that's a drug that acts on one of these receptors, NMDA receptors. We can do this, infuse this in the mice too. How do we know? So I do the same thing. Train the mice in the context, take two groups, one just a vehicle, saline, and CPP. The saline CPP is a drug that actually blocks this NMDA receptor mixed with saline. What do you see? Beautiful freezing for those persons who got the saline, but the other guy do not even remember. I put them back. It acts like, okay, I mean, what has I got to do with this context? Okay, now that's very good. Okay, and this is just telling you how deep can we go into understanding memory, right? At the molecular terms. Now, let's do this experiment. We took this and then switched to the different context, right? B context. You would expect in the B context, again, if you block with CPP, you shouldn't see any learning, while on the other hand, the saline should do. It turns out, if you are actually using a B context and B context alone, you still see the memory, learning happening. The reason why it is happening is because there is a previous learning. On top of this learning, you are able to acquire this new information independent of the molecule that we're talking about, NMDA receptors. Okay, now this again, a way for us to define what is a related memory. If I do this with the C, you don't see it. You will still see a deficit. Okay, now one of the criteria for creating uncertainty, right? Being able to define what are related memories that are related to each other. Okay, that in mice, we are good here. Can we say that we can create that? I hope so, right? Yes? Okay. All right. So you can also define it like memories that normally require NMDR activity, but can form without it if there exists a prior experience. We call that memories as related memories. Okay, good. So once you created that, Ivan Pavlov comes. Okay, we know this person, right? Time immemorial. When we talk about learning, when we talk about memory, we hear about Pavlov and his dog, right? What we do. I'm going to go through this slide. I can almost be sure. I mean, even today I teach in the for my undergraduates and graduates, and I tell them that no matter how many times you have heard, I'm almost sure, 80 to 90% sure, you have heard it wrong. You learned it wrong. Let me tell you why. Okay, so we say that okay, there are stimulus. Stimulus is things like food, external inputs. When that can actually elicit a native response, that can have a native response, right? It's wired, we are born with that, right? So these kind of stimulus called as a unconditional stimulus, and the responses that they elicit, we call it as unconditional response, okay, native response. But he also discovered there are also other kind of stimulus. I have shown a bell, but actually he used a metronome, okay? So what the point is that you can actually play this sound from a sound instrument along with the food, and sound per se in this context, okay, in the different context they may have a different meaning. In this context does not have any meaning to the dog, okay, it does not produce anything until now. But the moment you start coupling with the unconditional stimulus such as food, they make an association such that later on, if you put the CS alone, play the CS alone, you can actually see the salivation. Until here, there is no issue. The issue is why, when people try to explain why this happens, the immediate impulsive explanation is to say that the dog develops an expectation, or the sound, the bell develops an expectation. The bell has been heard, so my food is going to come. Okay, there you are implicitly meaning the dog is thinking about the cause and the action, right? It turns out that is anything but true. No, that is not happening at all. Instead, the dog is actually thinking the bell is the food. This is very different. Okay, it's not thinking that the bell is going to predict the food. No, I mean, bell is going to give its food. No, it is thinking the bell is the food. Okay, there are many evidences. Again, I can go down to and tell you, but that's the key here. So you develop some conditioning responses which are not thought-provoking at all, but that can happen. Pavlov investigated a lot of invested a lot of time, and discovered many such intricate details. And one of them that we are going to use is what is called as a higher order condition. Okay, why I'm taking a detour is because I'm going to use this related memory and the higher order conditioning to create uncertainty. Okay, so let's look at this a little bit more. Okay, so the kind of conditioning that we talked about here is called as the first order conditioning. Why? Because the stimulus that we are talking about, where we are importing a new learning or a new knowledge, is coupled to the unconditional stimulus direct. So it's a first order. Okay. On the other hand, this is very prominent. Okay. Again, it's very huge practical value. The prominence of second order conditioning can be understood very nicely here. Okay, we know that CS1, like a tone, coupled with food, can actually elicit a reward, right? There is no problem here. That just now we saw what that is. Now, what it turns out is that you can actually use this response once it develops, right? It's not a very thought-provoking response. No? It's just a conditional reflex kind of a response. Because it's a reflex kind of a response, what you can actually do is you can actually take this stimulus that is eliciting this response, couple it with some completely new one. Okay, so it could be another light. So this could be a tone, this could be a light. So the tone, you press do the thing, then you say the tone predicts food. Okay, tone, I play the tone, you get the food. Now you can actually do light and tone. The animal starts to treat the light as being equivalent of the food. Again, this is not some esoteric example, because if I just replace this with a token, okay, I'm going to replace the CS1 with token, so that what it tells you is a particular token predicts a food. Okay, the particular token is going to elicit a particular a characteristic response. Okay, you will do some work. Okay, the response here is you will do some work. Now, what you can actually do is that I can make you do something for this token. If you have not figured out this already, the token is nothing but money. That's what we're working. Money is not going to feed you, directly going to be of any value to you. Think about it, right? It's only because it can fetch you something that is of direct implication to you, right? How you develop and you start to work for money, not for the food and not for the other basic things, is very ingrained in our brain. Okay, all you are doing is that you start to forget the primary relation. You're doing this, right? This kind of relationship, development of this learning, is called as a second order conditioning, because there is no direct connection between the token or the money and the reward that you're going to have. It's indirect, correct? So second order, or we also call it as higher order association conditioning. This is very, very crucial, because as animals, when we evolve to draw correlations between so many different things. Okay, and it's very crucial for survival, and it helped. And so this aspect or this property seemed to have survived. Now, okay, so this second order conditioning, on one hand, and our ability to create these contexts A, B, and C, right, with specific relations. What I have done here is that think of this box as a global set of all the features that it can think of, and I have carved out a small subset and called it as A context, another small subset called it as B context, another one called it as C. Now the overlap tells you how much of the commonality exists between A and B. And the way I have drawn it, you can actually see very little exists between A and C, but through the B, A and C can be linked. Okay. Now for this linking in to happen, A to C, you need a higher order conditioning, because it comes through B. What I'm going to show you is that such a conditioning can happen when there are uncertainties. Good. All right. Okay. So let's go back to our mice and the fear conditioning. You take the mice, place them in context A, provide them a mild food shock, go ahead and test them in B, A, and C. When you test them, you get this now pretty much the same data, exactly the same data. It's not exactly the same, pretty much the same. The only difference being that there are much more animals here because of things that we are going to do next. Okay, much more animals here. So you can see that the B and A are kind of 30 days later we are checking, right? So they cannot tell apart where they have trained. So they don't differentiate. But then if you put them in C, there's a humongous difference. But what one of my grad students, Aditya, what he did was that, hey, look, after 30 days, granted, I cannot tell if it is B or A where I trained on. But what I can do is that instead of testing them in B first, okay, a context that does not, that is not where I have trained, I put them back in A. That kind of serves as a reminder, you see. Okay, then I put them in B. If it is true that I am unable to remember whether it is A or B, what is going to happen is that A, by me putting it A, I'm going to kind of shook the animal to say, okay, now look, there is something fearful that even that has happened here some long time ago, but yes, it has happened. When I put them back in B, okay, what's going to happen is that again, this is a immediate happening. That is, I don't know if it was similar to 30 days before or not. Yes, I know this was a fearful event. It was fear inducing, but I'm not sure if it is this or that. Okay. In this scenario, the animal does not have that opportunity, because when it experiences this uncertainty, there is no fearful event for it to afraid of until that point in time. Okay, so now that fear, what is going to happen with that fear, what it can actually do is that it can transfer the fear, the conditioning that is associated with the A to B, and you can actually test it when you are actually overlapping it. C, and that's what happens. First and foremost, they differentiate because the A acts as a reminder, and you compare the C's. Okay, the green bars here, they're pretty different. It's actually marginally high. It's marginal, but still, it's significantly high. You can actually see it, and that's very, very alarming and disturbing, because this is a context that the animal knows for sure is distinct, and it has never experienced, never seen it before in life, and starts to develop a fear just because the way in which you actually retrieve the memory. You have created a fear for an event that never has happened in its life. Okay, so that's behavior. Whenever we get this kind of a behavior, many times it's interpretation of an interpretation, right? Why is that? Because the animal is interpreting what things are happening around it, and you are interpreting what the animal is interpreting. Okay, that's always a problem. You can replace the animal with the humans, is still the same problem. Okay, so as a neuroscientist, you would like to get to the neurological basis or a neural correlates of it. The way we do that is by asking, are there any brain regions that are responsible for encoding this? Okay, so all of this fall under a category of memory. There are many different categories of memory. One of the categories of memory that we are actually talking about, event and facts, that I started with, is hippocampal dependent memory. So we asked, okay, do they form enduring unreal memories? That is to say, just the way you or that the couple remembered that there was a boat, they will remember that this was an event that for their life. Even if you remove the hippocampus, so we said, okay, if you remove the hippocampus, do you still see this memory happening? The answer is, you do need the hippocampus to form this memory, right? Okay, if you lesion out the hippocampus, obviously you cannot form this higher order association. That's not big a surprise. I mean, it's a new finding, but it's not a big surprise, because you need to have this event, right? Is that the okay? Hey, look, something bad has happened to me. Okay, so that that memory needs to be there. So it's dependent on hippocampus. But what is surprising is, once this C memory has gone up, you can actually lesion out the hippocampus. Okay, I'm sorry, it's not coming out here. It's a lesion is by this scratched out thing, but this is a sham lesions. So what you can actually see is that they are actually pretty different, and saying that you need this hippocampus actually to form this memory. If you lesion out the hippocampus, you don't form this memory, but once you form it, okay, what you can actually do is that you can actually lesion out the hippocampus. Okay, so 33rd day is when we actually, if you remember the actual training paradigm, right? So 30 days wait, 31, 32, 33, 33rd day is where we are putting them into a completely new context and saying that, okay, they have formed this unreal event, and that unreal memory, okay, you lesion out and test like 24 hours later, you lesion out, or 30 days later, you lesion out. You test them on the 41st day or 66th day, they're pretty nice and intact. Okay, they not only form an enduring memory, but they form it very fast, right? You had to wait 30 days to get this. Okay, here 24 hours. 24 hours is all it takes. You can lesion out the hippocampus, the memory is still going to stay there. They not only form long-standing memory, but also okay, good. Sure. They also form rapidly. Good. Now, um, it's what is it driving? So the driving force is because of the conflict between, is it really A or B where I'm trying down, right? So from our different line of research in our lab, we also know that a specific class of neurons, okay, we are not now going deeper into not the brain regions, but we are going into the specific kind of neurons. In the brain, there are different kind of cells, and the, one class is the neurons. In the neurons too, there are different classes. Inhibitory neurons versus excitatory neurons. In class of an inhibitory neuron called as a parvalbumin neurons, abbreviated as PV neurons, you actually shut down the PV neurons. We know that our ability to tell the difference between A and B is getting affected. So what we do is that, how do we shut down is we generate virus particles, okay, the virus-like particles. It's not virus. I mean, we make use of the machineries of the virus, but it cannot multiply, okay. So we make virus-like particles that can actually go and transfect a class of neurons and transfect and express ion channels in a class of neurons called as a parvalvular neurons. The idea is that they express a particular protein called as a designer protein, a designer protein, and they are can be activated by a designer drug. So there is no endogenous ligand to it. So that's the advantage. So what we did is that you can actually express them in PV neurons. CNO, clozapine N oxide. Clozapine itself is a psychoactive drug. By being in IIC, you can actually collaborate with the organic chemists, and they can actually synthesize the CNO quite nicely, and they are non-psychoactive. Means, and but then they can actually go ahead and inactivate the PV neurons. When you do that, what you see, okay, when you do that, so that's the evidence for you to say that, okay, they are existing. But the when you do that, what you see is the suppression of the C. That just insert shows you that if you don't activate, you don't suppress, that's where the C should have been. But because of the CNO, you are actually able to inhibit this conflict creation or the uncertainty creation, thereby inhibiting the memory formation itself. We can even go further down to the neuronal level. Typically, when we talk about the memory, there are different scales, either at the synaptic scale or the molecular scale or the pan-neuronal scale. And thanks to Cahal, age-old, 1896, where he kind of said, okay, look, it could also be because of the number of these synaptic connections that can change the spines. But in the lab, we were fortunate to stumble upon a new phenomenon, a new kind of plasticity. We call it as a clustered plasticity, wherein the numbers doesn't change, number of the spines, okay, number of these synaptic connections doesn't change, but rather something else happens. You start measuring the distance between these two, and you see the per unit length, the number of such protrusions are the same. Think of that as spines, but the local density starts to increase, or clustering happens. So because it's related memory, we were actually asking if such phenomena could actually express or could explain what we see. So the way, in order to do that, again, we go to the mouse and we surgically implant cover glass slide and look into their brain. That's a small reconstruction. I mean, that's a reconstruction of a small portion of the brain, actually. And the advantage with that is that you can actually study with very high resolution these synapses. What Cahal was doing it in dead sections, you can actually do it in live mice as a function of learning. And with that, what we could actually see is that if you measure the interspine distance in the group that actually express the memory, you see that the distance coming closer and closer, while the other group does not stay there, does not consolidate. Okay, so that's kind of bringing into one part of the story. Am I good with the time? Okay, so I think I would just stop there, but then I have to kind of show you the people who did this work, okay? And otherwise, but so that's our team, reasonably small. Nirupam, Manish, Nirupam did much of the work on parval women interneurons. Manish, his work I did not talk about, but he did some of the activation work. Aritra, Sriji, Rajesh, and Saraj are in the lab, and our funding comes from these sources. Thank you all. Ready to question.
Thank you so much, sir. The way you explained the uncertainties of our memory and how memory works over time through scientific investigation and research works was very comprehensive. And I hope that everyone has learned something from the very wonderful and comprehensive presentation given by sir. Thank you so much. We will proceed with the question answer session. We're keeping it a little short because we have the next speaker online waiting for us. So it is scheduled. So I would like to open the conversation for the audience. If you have any questions, you can ask Professor Balaji.
Just out of curiosity, what is the sample size? I mean, how many times has been the
Which one?
my sample?
Well, quite a few experiments, right? So each of them 20, 40.
Cohort.
Yes.
Any specific reason why all this happens?
Yeah.
I
But also the genetic transformation that I was talking to you about, and that can be it's not important nowadays.
I'm aware of situations, as I was saying also.
And
Because people say that humans are award from monkeys. So when there is proof that there is wrong, so it's could be one of the.
well, I'm not sure if I would believe that it's wrong. No? Second thing is that these mechanisms very much concerned kill the mouse. So I so it's easier to do that. There is also a ethics issue.
So do you sacrifice this? Do you sacrifice the sample studies over?
Yes. We do.
Said in the hello. Okay. So, sir, you said that in the Pavlov's test, the dog thought that the bell is itself food, but olfaction of dog is very strong. So, when in the unconditional stimulus, the food is given. So, yeah.
Yeah. So
Yeah.
I get this question every time I say, okay, I know my dog, my pet is very super intelligent and all that, right? So if I prove, I can be stupid, right? So humans can be like that, right? Reflexive. You will not ask that question, perhaps, right? Look at this example. We all go to higher experience, right? We want to go to higher floors. What we do, we go near the elevator. Right? Most often we have to wait. There is a button on that. You press it. Okay. Here a light comes on saying that you have pressed it. The elevator takes a little longer time to come. We know that we have pressed it and there is feedback, right? Do we stop pressing it? Keep pressing it. It is just that. It is just that. The dog, I mean, if you consciously think about it, yes, it's true. You will know that. No. Perhaps next time you go to the elevator, you won't do it because it's there. But if you had not done that, This is our very inborn reflex. Came from there. Another example. Not prevalent that much these days, given the cell phone synchronization. But previously, my fathers and grandfathers, they would used to set the time of the clock to 5 minutes past. Think about it. How rational can it be? You set the time to be 5 minutes faster so that I can know that I will be on time, right? It doesn't make any sense at all. Think about it, right? You are the person who is setting it. When you look at the time, don't you think that you will subtract that 5 minutes and go? No, because they are hinging on the fact when I look at the time and going, right, there is not much time for me to think. I don't spend the time, I don't spend the cognitive effort to think. I look at it, act on it. Reflex. That's exactly what the dog is doing.
Thank you, sir.
Yes.
Professor, thank you for the lovely presentation. I have a question on that Pavlovian response that you mentioned. You said that you introduce another token, which can be money, which can be misinterpreted by your brain as something to do with. It's a way to get food, like the dog does with the pet. So in a real life scenario, my question is, as you know, Bhaktivedanta Prabhupada used to say that our need for money, okay, also is not only about survival, but also about the other aspect of reproduction. We want to expand, and that's why we want to build wealth. So is it how we are differentiated from a dog? The dog looks at it like typically food, but we look at money also not as food, but also as other aspect. Just a question.
Yeah, no, good question. So you can think of this, you can replace that food by any things that helps for survival, okay, in general. But for us as humans, right, as we move up, we start to equate. There are, I mean, this is one example I told you is the second order, right? That's like that. You can keep building higher and higher order. That goes way deep, and then we lose the track of why we are doing what we are doing. Okay. By the way, this is holding good even in chimpanzees and primates, where actually they have done an experiments where they will train a monkey to get a juice or a grape for a token. They have to do something for a token, and once you do that, what the observation is that the not only the monkey start to work for the token, gain the token, but it starts to, if there is a dominant monkey, right, it starts to grab the token from the other person. Okay, so that's very much ethological.
Thank you, Professor.
Thank you.
Sir, it was very nice to hear you talk. And in that experiment where we used CPP as a block of NMDA receptors, we still found that if the organism was primed by a similar context, we found that memories can still be formed. So has any work been done about the alternative mechanism other than the NMDA receptors which are involved in memory?
So there are multiple directions people have taken. In our lab, what we had taken is in terms of clustered plasticity. I did not talk about it, but what we think is that the memories that are related, right, when you're trying to form, they do not require the entire ensemble of things starting from the molecular basis to form. But in fact, they can make use of what is existing already, okay, and build on that. Okay, it's you can think of this as kind of an energy conserving thing. Like if you people have seen that whenever there is a new synapse that are formed, it draws its energy from the local mitochondria that is present. And if you deplete that, you won't see it. But once you have that mitochondria already present, the probability that the next synapse present next to it, very close to it, will encode the plasticity or encode the memory is very high. So things like that, people have done it. So we are moving along that lines. People have also looked at other channels, I would say that that's not that very successful in that.
Sir, thank you very much for the wonderful talk. And I would just like to know what is the direction? What I understand you're trying to understand how the memory is working. Is there a specific direction you are looking to achieve it after this research? What you are ultimately trying to achieve?
Yeah, that's a big question. But general theme of the lab is trying to understand how our brain is using this learned experiences to arrive at solutions that are novel almost instantaneously when you're faced with a new situation. So we have done some behavioral experiments along that too. I did not talk about them. So again, just the way we humans try to solve like a Sudoku puzzle or a chess puzzle, right? There is no representation of that solution exists in the brain until that point in time, but you are able to, the brain is able to come up with it. But the way in which it can come up is very different from whether you have solved 100 puzzles or no puzzles at all, right? So similarly, we can equate things in mice and do that. I would be happy if I um, and find some of these answers by the time I retire.
Thank you very much, sir. Thank you.
Thank you very much.
Yeah, thank you. Thank you so much, sir, for the wonderful presentation and the invigorating session. To felicitate Professor Balaji Jayprakash, I request Bhakti Swaroop Rajendra Kumar Swamiji to come up on the dais.